Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we will interview one or more authors of the feature articles, the papers of the month, or other articles published in that month's print issue of the Diabetes Journal. My name is Darleen Sandoval, and I'm joined by my podcast co-host, as always, Dr. Kirk Haberger and Dr. Kevin Williams. How are you all doing today?
Hey, guys.
Hey, doing pretty good.
All right. So we have two internet celebrations this month. I don't know if you knew about this, but it is Women in Medicine Month. And it is also National Childhood Obesity Awareness Month, which we'll be doing something special focused on that next month. But I thought it was kind of interesting that next month is also followed, we're following National Childhood Obesity Awareness Month by October 4th being National Taco Day.
Ooh, of the three, I'm taking Taco Day.
Yeah.
Yeah. Is it followed shortly thereafter by Burrito Day, or?
I didn't see it on the list of October internet holidays.
Uh-huh.
Other internet holidays y'all are aware of that we should be celebrating?
I did a brief search and I saw today is National Lazy Moms Day.
Oh, wow.
So for all the moms out there, this is your day to take it easy.
Is it only for the moms who are lazy, or is it to allow moms to be lazy? I'm a little confused.
Yeah,
I'm not wading into those waters.
Yeah. I'm not a mom, so I'm a dog mom, so
Absolutely.
But my border collie does not allow for lazy mom days. So she will be getting her run as per usual.
Yeah, I don't think anybody would accuse you, Darlene, of being lazy. I think you're
Kirk, you're busy in your household this week with homecoming. Is that what you said?
Oh yeah. It's homecoming season. We built a float, and then we had to tear said float back down. So it's been a week already, but that's all right. It's a lot of fun.
How about you, Kevin?
This week, I really don't want to talk about what happened last night, which was a game that occurred of the beginning of the season. But it's interesting to get those things started back up and see what happens for the rest of the season. It's always, your own team's year, right? This is the year for them to succeed. And then that first game happens or whatever, and everything goes downhill from there. At least that's my history.
So I don't know if you know this, Kevin, but your team is the team that I have hated. I've been raised to hate the Dallas Cowboys.
Yeah.
My dad would disown me from his grave if I rooted for the Dallas Cowboys.
So what was his team?
So we were big Joe Montana fans, so the Niners and then the Chiefs.
Yeah.
So yeah.
Well, the Chiefs is another Dallas team. They started off in Dallas before they went up to Kansas City. Yeah.
Oh, I didn't know that.
Yeah.
All right. Well, I guess we should talk some science. Kirk, we have a obesity drug themed episode today. You want to tell us about the papers we've discussed?
Yeah, absolutely. So the first paper that we discussed this month was titled GLP-1-mediated targeting of inflammation corrects obesogenic memory in male mice. We chatted with co-corresponding author, Dr. Carmelo Quarta, about his work, and it's always fun to catch up with people that you've known from prior time. He was a trainee when I was a trainee, when you and I guess all of us were at Cincinnati, and it's great to see him have some good success there.
Yeah, for sure.
We then had a chance to talk with Dr. Willa Hsueh. She's the last and corresponding author of September's paper of the month, which is titled GLP-1 receptor agonist Dulaglutide attenuates hepatic steatosis on obesity via a weight-independent mechanism. We had some very cool data that leaves us with still some questions. How do you get such direct effects from a tissue that doesn't express the receptor you're agonizing, right? So that was a lot of fun too.
And Kevin, we have another fun Sweet Talk episode.
That's right. Our guest today is Dr. Alexander Nectow from Columbia University. And he's asking a simple question of how does the brain know when to start eating and when to stop? As a Pathway to Stop Diabetes awardee, he's been able to follow that question into new and exciting areas, and the answers are offering new insights into not only body weight control, but also risk for diabetes. We're excited to share his story today, so stick around and find out more.
All right, great. Let's jump in.
This episode's first paper is published in the September print issue and is titled GLP-1-mediated targeting of inflammation corrects obesogenic memory in male mice. Today, we're joined by last and co-corresponding author, Dr. Carmelo Quarta. Dr. Quarta is a principal investigator at INSERM, which is the French equivalent of our NIH in the US, as well as a group leader at the Bordeaux Neurocampus at the University of Bordeaux. Dr. Quarta, congratulations on your paper, and thank you for being a part of this month's podcast.
Thank you very much for the invitation and for organizing this.
Carmelo, it's so good to see you. We like to start with a icebreaker question. So we didn't give this to you ahead of time, so a little bit of a surprise. But can you provide a fun fact about yourself that has nothing to do with science?
Oh, that's quite a nice nice breaker to me. I was just discussing time ago, with colleagues, what would have been my possible job if not being a scientist, and I had quite a hard time of defining one, and I ended up that I wanted perhaps being a policeman. I don't know if that's fun enough, but I'm used to kind of be a little bit the policeman of the lab sometimes because I like having things in order, and then I sometimes I'm paranoid about having things like under control.
Nice.
I don't think there's any wrong answer to that question, right?
That's right.
All right, Carmelo, before we discuss your article proper, we always like to give our authors the opportunity to highlight any other contributing authors or collaborators that, made a difference in this work.
I would like to mention that, this work was beautifully led by my PhD student Stefan Leon, and is actually co-supervised by Dr. Daniela Cota. This is the result of a team effort where we had several scientists at the Bordeaux Neurocampus contributing, and also external scientists. And among them, I have to mention and acknowledge Brian Finnan and Bin Yang, who are the major chemists that have produced these famous bullet drugs that we have used to target inflammation in our obesogenic in models of obesogenic memory. And last but not least, this work starts really a little bit from my postdoc work at the Helmholtz Diabetes Center in Munich. And there are a lot of, great scientists and the folks that I think you all know that have been really the first to design this idea of generating molecule where you can, target GLP-1 together with nuclear hormones. And, among them, I have to, of course, cite Matthias Schoep, Richard DiMarchi, Timo Müller, and Christopher Clements. And, I wouldn't be here telling you this story without their previous work.
That was a great introduction. Can you maybe give us just a little bit more of an overview on your research program now, and then how this specific project grew out of it?
We work in a neuroscience institute, and our research focus on understanding how the brain communicate with peripheral organs and the other way around to control metabolic pathways, but also energy handling in our body. So how our body can eventually store or dissipate energy, and how changes in this communication process relate possibly to the development of cardiometabolic diseases such as diabetes and obesity. Within this major research axis, we became interested in understanding a question that we think is not poorly understood in the field: why people suffering from obesity have such a hard time to lose their weight and then to maintain the weight lost, and why then they eventually regain the weight lost over time. So what is generating and creating in them this kind of what we call obesogenic memory? And that's how we started this project, by thinking and looking at the literature and seeing that actually inflammation seems to be a big deal, in our opinion, in mediating this kind of obesogenic memory. And therefore, we thought it would have been possible and perhaps nice to think about, precision pharmacological strategies to target this obesogenic memory by targeting metabolic inflammation.
Yeah, that leads really well into the next question. I mean, we know that the new GLP-1 targeted drugs are having clinical success and of course media success as well. Um, Given that backdrop, tell us a little bit more about the rationale for why this current study is so important.
Yeah, I mean, these are exciting times, right? So we're all excited in the field. We have these molecules that are doing exceptionally fine, helps people really, improving their metabolic health and losing body weight. Everything is really nice and exciting, but there is one aspect perhaps that we need to discuss a little bit more and then to be aware of. Even with these molecules that have undoubtedly beneficial effects, we are not really solving the problem in the long term, because as soon as you stop the treatment, what we observe is that people will eventually regain their body weight and the metabolic disease will return, right? So even with the best molecule we can have now, we are not really addressing the root of the disease and why actually there is some kind of a signature of the disease that is staying in the body of these people. And they try to do all they can to actually have the disease coming back after you stop the treatment, right? They will eventually regain body weight. So, and then we started to think, how can we improve that? And how can we make better GLP-1 molecules that could have more longer term effects in terms of body weight maintenance and potentially address a little bit better this problem of the obesogenic memory, the fact that the disease tends to return all the time. And that's how we thought that there were room for improvement by combining GLP-1 pharmacology with an anti-inflammatory agent. Why? Well, because there is quite a bunch of literature telling that in response of during obesogenic conditions, there is a state of low grade chronic inflammation that seems to be somehow linked with the fact why people defend their higher body weight and have hard time to lose weight. And it seems also to mediate why certain people can eventually regain the weight lost after a whatever weight loss intervention. For example, there are studies showing that even after bariatric surgery, you look in the fact after long term, these people have lost weight, but you still see signature of inflammation in adipose tissue that stays over time. And also you have studies telling you that this signature of inflammation, for example, can predict whether someone is more or less prone then to develop weight regain and to have the disease returning. So we thought it was somehow a smart idea to try to play with GLP-1 and inflammation to kind of address a little bit better this problem of the obesogenic memory. And that's how we came with this idea of testing this molecule where GLP-1 is conjugated with an anti-inflammatory agent, an immunosuppressive agent that we all know about, dexamethasone.
You've mentioned obesogenic memory in the context of clinical observations, and you've developed a mouse model of that state. Could you kind of explain conceptually what that really means down nuts and bolts on obesogenic memory, and then tell us a little bit more about that experimental model?
Well, I think obesogenic memory, I realize it's a quite a broad term that I think can include many different biological processes. Overall, the way we interned at least obesogenic memory is the ensemble of those biological pathways that somehow programs subjects suffering from obesity to always defend their elevated body weight at all cost, right? So we know that those subjects, as I explained before, have a hard time to lose weight, and they eventually tend to regain the weight loss. So this is for us what the mesogenic memory is. It's a little bit a combination of mechanisms and biological processes that explain two different components. On the one hand, why after you're obese you have hard time to lose weight, and on the other, why then after you lose weight you can be in most cases prone to regain the weight loss. So these two components to me together define the concept of, or at least the concept that we have in our mind of obesogenic memory, this memory of the disease. And it's important to remind and note that in this mouse model that we have developed, we are addressing only one part of the problem, which is why and how people with obesity, or actually mice in this case, defend their elevated body weight, right? So what is making them to remember that they're obese, and they have to always have a different body set point compared to an animal that has not been exposed to an obesogenic condition. How does the model work? But it's quite simple. So basically, we have used adolescent mice, 4 weeks old. We expose them to an obesogenic diet, a high-fat diet for about 3 months. And then after we give them this diet, the animals gain body weight, they gain fat, of course, and then we switch them back to the same chow normal caloric diet that they had before. And then when we switch them back, what happens is that they will eventually at the beginning lose a little bit of body weight, but they will never reach the body weight of control animals that never had, the obesogenic diet and that were fed with the regular chow diet all the time. And this for us suggests really the establishment of body weight defense mechanisms and therefore the establishment of some kind of obesogenic memory, because the animals never reach the body weight of its control, even after several weeks after they've been switched to the normal chow diet. And while we think this model can be helpful also for looking, for example, at the link with inflammation that I mentioned before, well, because in this model, we observed that even after long term of the switch to a normal chow diet in mice that were exposed to one episode of obesity, we have changes in inflammation in the fat. We have signature of inflammation in the fat and also in other organs. And this signature doesn't go away. I mean, even if you, for example, use calorie restriction to push the body weight to these mice to really go down to the level of the control in chow diet, even in that case, you have this kind of signature of inflammation that stays in the body of these animals. And that's why we think for us is a good model to also look at the link between inflammation and obesogenic memory.
Good. And I guess just as a quick follow-up, maybe can you tell us a little bit about the GLP-1 dexamethasone conjugate? I know you've published on this in the past, but what is it about this peptide conjugate that allows you to avoid some of the systemic glucocorticoid side effects that we are well aware of?
Yeah, so this is, of course, a very important question. This is basically a unimolecular conjugate where you have two parts of the molecule, a GLP-1 part of the molecule and another part that is the dexamethasone, that are kind of chemically fused together by a disulfide bond that is basically attaching dexamethasone to the C terminus of the GLP-1 molecule. The result of this chemistry, magic chemistry, developed by Richard DiMarchi and Brian Finan in collaboration with my old lab, as I said, and Matthias Schoep, is that the molecule is supposed to be quite relatively stable when it's outside the cells, right? So you have the two components that are not supposed to kind of be broken up. But then as soon as the molecule is internalized by cells that express the GLP-1 receptor, then you have a two mode of action. So you have kind of a Trojan horse approach. On the one hand, you activate the GLP-1 receptor in that cells, and on the other, you're going to have a chemical reaction that will break the bond between the GLP-1 and the dexamethasone. And therefore, dexamethasone can be really liberated into the cell and act and induce intracellular signaling by, for example, activating the glucocorticoid receptor. So you have quite a specific way to address an anti-inflammatory agent into a cell expressing GLP-1 receptor. And if a cell does not have the receptor, then it's not supposed to be targeted by the compound. And what is the advantage of this? Well, so the advantage is that this way you can bypass some of the effects of dexamethasone that you would not want to have for an anti-obesity therapy. Because, for example, we know that dexamethasone in the fat might, by activating the glucocorticoid receptor, induce adipogenesis, makes more adipose cells. Or in adrenals, it would, play and alter steroidogenesis. And so you could have effects on the HPA axis that you don't want to have. Or in the liver, it could induce gluconeogenesis. It could induce higher production of glucose. And those are also things you don't want to have, And those are tissues that we know they have, negligible expression of a GLP-1 receptor compared to other target sites. So by this way, we can really, address the molecule where we want. For example, in the pancreas, where we know it's one of the target of the molecule, the hindbrain, we believe also, of course, the brain, and also we believe that there is something going on at the level of the immune cells, because, there is now some increasing line of literature telling that GLP-1 receptor can be expressed also in certain immune cells, which I think are also, one of the target of our pharmacological effects, although admittedly, we have not directly investigated that yet.
Great.
So you treat the animals for 7 days with the conjugate, and you find that the CCR2, CCL2 inflammatory pathway is a specific mediator of the glucose intolerance and adipose tissue inflammation that links to the obesogenic memory. Can you talk a little bit more about the role of these monocytes and how they're playing a role in memory?
Well, actually, this is a beautiful question, but I'm afraid we really don't know the answer yet, right? I mean, this
Okay.
is part of the mechanistic studies that we would have loved to do, but for question of time and because we really wanted to show this, as early as possible, we didn't investigate. But it is an interesting finding, I believe, because for a series of reasons. Well, we know that this ligand, CCL2, that we see increase in the fat of our mice after obesogenic memory, it's important for recruiting macrophages in adipose tissue. And macrophages expressing, CCR2 and monocytes expressing CCR2 is likely important for contributing to the inflammatory reaction we have, we observe in adipose tissue in our model of obesogenic memory. And what we observed is that with our conjugate, we restore this elevated CCL2 signaling in our model. So this could be one potential mechanism that explains why with our molecule, for example, we see an improvement in adipose tissue inflammation in this model. So why is this interesting? Because I think, well, on the net of the fact that we still have to demonstrate whether really, changes in CCL signaling in certain organs mediate the pharmacological effects. We don't know. But this is giving rise to me to the interesting idea of further exploiting whether co-modulation of GLP-1 and the CCL pathway could be a good way in the future to really have molecules that are quite potent in terms of correcting perhaps metabolic inflammation in obesity, and perhaps why not also improving this problem of obesogenic memory. And this is, I think, a good or a possible scenario for the future, because we know there are clinical studies where CCR2 and CCR5 antagonists have been tested for the treatment of type 2 diabetes and for their anti-inflammatory effects. And I think it could be interesting to just explore further whether combination of these molecules with GLP-1 could somehow have beneficial effects in terms of long-term body weight maintenance and metabolic control in obesity.
So Carmelo, I have to ask you this question. As I'm reading through your paper, right, you describe some great benefits to glucose homeostasis, but no changes in canonical GLUT4 expression, right? I guess, and you looked at mRNA. Do you have any idea how you're benefiting glucose homeostasis systemically, but not really changing insulin-mediated glucose dynamics, if you will, into the adipose tissue?
Yeah, so we don't, but there is one possibility, which is the fact that what we observe in this model, and after, for example, treatment with GLP-1 dexamethasone, is some kind of amelioration in the activity of brown adipose tissue instead of not only in white adipose tissue. So we know that when you activate the brown adipose tissue, you can have effects in terms of controlling blood glucose and also glucose uptake in several organs, at least in mice, right? So, and of course, one could speculate that this could be one alternative pathway, activation of thermogenesis and glucose uptake by, in general, increased brown adipose tissue activation could be perhaps one possible mechanism. But you're completely right. This is clearly an open question that, deserve way more studies that we haven't been able to do that yet, unfortunately.
It was a very cool finding.
Yeah, thank you.
So I have to ask about if you expect any sex-specific differences in obesogenic memory or the inflammatory responses or even responses to this drug.
Yeah, this is a very important question. And of course, we know that, GLP-1 pharmacology is effective in males as in females, which is a good news. But I think what we know that, of course, metabolic inflammation is equally important for, inducing metabolic disturbances in both sexes. So, of course, now there is a lot to be discussed also in female, especially, before menopause and after menopause, where the contribution of inflammation may be different, of course, in relation to obesity. And, the key, let's say, difficulty, but also somehow room for further studies is to establish, in my opinion, a model of obesogenic memory in female that would work the same as in male mice, right? Because for example, in our model is based on the exposure to an obesogenic diet for 3 months. So the animal is reaching a certain weight. Of course, we know that if you take young mice, as we have done here, young female mice, and give them an obesogenic diet, they're kind of resistant to the obesogenic diet. So you have to really modulate that kind of protocol. So this requires some troubleshooting that I think it would be worth really doing. So to really create a better model of mouse model of obesogenic memory, that in my understanding, I'm also looking these days a little bit in the literature, there aren't many models in female to really look at obesogenic memory. So I think it's something that we are all expecting in the field. So I don't know what the prediction is. I think the prediction is that there is no prediction. We should really try to do the experiment in female and see how the compound works. So, but I would expect to see some surprise. based on whether you kind of play with obesogenic memory before, in young mice versus aged female mice. And I think this is also an important thing to consider in this follow-up analysis that we should do in the field.
Carmelo, are there any other key points or thoughts that you'd like to leave us with, or maybe give us a hint about where you think this project might be headed?
Well, I think what we hope this project will be headed to is to really follow up studies addressing in the field what are the main organs and cell types that are really susceptible to undergo deregulation in response to obesity, and how an episode of obesity is somehow what are the main cell types that are more important in programming, for example, the body of people suffering from obesity to kind of defend the elevated fat mass accumulation and to have the disease always returning, right? I mean, what are the cell-specific mechanisms responsible for this obesogenic memory? This is a key question for explaining our findings, because our conjugate is targeting GLP-1 receptor cells, which can be found in many places of our body. So we don't know which one is really, which organ is really mediating what we find. And in general, for our basic knowledge on why it's it's so difficult to have people losing weight in the long term and having them actually sticking to this even when you actually unleash the treatment, right? Why the disease is returning? What is the main cell type more important for that? These are open questions. I would be, excited in the future to address more, study more the role of the immune system. And we have some idea. We think that, for example, regulatory T cells could be particularly important for that, for a series of reasons. I mean, well, we know that some of them actually have the GLP-1 receptor. There are preclinical studies showing that if you, for example, play and try to deplete T cells, you can improve the way mice are undergoing this kind of yo-yo effect of weight gain and weight regain and weight loss. So I think there is a lot that if I would have a very big grant to invest on, I would really invest it in there and trying to look really how different types of immune cells may contribute to obesogenic memory, and how with GLP-1 pharmacology we could try to target that.
Very good.
Excellent.
I don't have the money now, so it's something that I would.
Yeah, if anybody's listening wants to. Dr. Quarta, we'd like to thank you for joining us today and sharing your expertise and discussing your recent work on GLP-1-mediated targeting of inflammation and obesogenic memory. I encourage the listeners to read the full article in the September issue of Diabetes.
Thank you very much. It was very nice being here with you, and a very nice initiative. It's been real fun. So thank you very much for the invitation, for everything.
Next, we're going to discuss the September issue paper of the month titled GLP-1 Receptor Agonist Dulaglutide Attenuates Hepatic Steatosis in Obesity via a Weight-Independent Mechanism. Today, we are joined by the last and corresponding author, Dr. Willa Hsueh. Dr. Hsueh is professor of medicine in the Division of Endocrinology, Diabetes, and Metabolism at The Ohio State University College of Medicine. Dr. Hsueh, congratulations on your paper, and thank you so much for being a part of this month's podcast.
Thank you, Darleen and Kirk, for inviting me.
We didn't send you this question, but we like to start with an icebreaker, okay? So I'm hoping maybe you can give us a little insight into who you are by telling us an activity that is not science related that you enjoy doing, in the real world.
I like to attempt to play golf.
Nice.
And it's more because I can be outside, right?
Yeah.
And enjoy the scenery as opposed to controlling where the ball goes.
Someone much smarter than me said, golf is always good because in the end, at least it's a walk in the park.
Exactly. That's my take on it. Thank you, Kirk.
All right, so now we want to talk a little bit about your research. So before we discuss the article, we like to give our authors the opportunity to highlight any other contributing authors or collaborators on this work.
The two first authors are outstanding postdoctoral fellows on my lab, Dardi Shantaram and Shalal Rima. The other real contributor is Dr. David Bradley, who was at Ohio State several years ago and is now an assistant professor at Penn State.
Nice. And can you start by giving us a little bit of more general overview of your research program and then maybe a touch on how this specific project fits into that?
Yes. My research for the last almost decade has focused on the adipocyte. We look at the adipocyte as a metabolic cell, as you know, and it controls a lot of metabolic functions like insulin sensitivity and delivery of fats and nutrients and other things, including microRNAs to target organs throughout the body. Now, when obesity sets in, or actually, it's not really obesity, even high fat diet sets in, the adipocyte changes its function. And instead of becoming a metabolic cell, we're studying how it becomes an immune cell. So we've published that within 2 or 3 weeks of high fat diet in humans or in mice, both, the adipocyte is capable of turning on the major histocompatibility 2 pathway, presents antigen, increases Th1 cells. And we think this is one of the reasons why the immunosuppressive or protective Tregs fall. And this sets off a cascade then of inflammation in fat. Now, this fat inflammation then, as you well know, affects every part of the body from head to toe. It's associated with dementia. It's associated with heart disease, both heart failure, coronary artery disease. It's associated with metabolic associated fatty liver disease. It's associated with type 2 diabetes, of course, arthritis, thrombophlebitis, and even cancers. So the whole body, every organ gets affected. So we've been trying to understand how the fat sends messages. And we found a lot of my work going on now is looking at how extracellular vesicles from adipocytes, what they contain, and we're doing multi-omic studies and looking at how they affect distant organs. So one of the issues that came up, of course, is when you lose weight, with what happens. And of course, the blockbuster weight drugs are glucagon-like peptide receptor 1 agonists. And, the whole world is on them, I think, or trying to be on them, whether they're fat or they're thin. I don't know. People want these agents. And so there's like almost a paper every 2 months in a outstanding clinical journal with a clinical trial showing their positivity. Now, every time you look at a clinical trial, especially the trials associated with cardiovascular disease, and it's in patients, they've been reported in patients with diabetes, with non-diabetes, there's a prominent reduction in both cardiovascular disease and weight. And so we asked the question, well, is this related to the weight loss? Because many of our studies, and we'll talk about our mouse model in atherosclerosis, the more the weight, the more the vascular disease. So we ask if there's prominent weight loss, is that the cause of the improvement of the obesity associated endpoint, or are there direct effects?
Well, I love that idea of the adipocyte switching from a metabolic cell to an immune cell, right?
Yeah, I have to write a review article on that someday.
That's brilliant. I love it. But we're going to talk today a lot more about the liver, right?
Yes.
And so maybe you can bring us up to speed on current strategies used to treat MASLD MASH and how we model that in mouse.
Yes. So there are a number of models in the mice for MASH. We use this LDL receptor knockout mouse. And when we age it, it gets a marked acceleration of atherosclerosis. The young mice, which people use that are only about 8 to 12 weeks old, have fatty streaks in their blood vessels. Whereas we found if you let the mouse go to middle age, like it's a year old, if you can afford the expense, the mouse gets accelerated plaque formation with cholesterol crystals, a lot of inflammation around these crystals, more like a human plaque in a coronary artery. So we used that mouse, and we also found that this mouse gets accelerated liver disease, and the liver enzymes in this mouse increase, as well as a marked deposition, of course, fat as or steatosis, as well as ballooning degeneration, which is a marker of inflammation, and fibrosis as well in this model. So we think, like humans, where cardiovascular disease parallels the liver inflammation, that this mouse resembles that. So that's why we use it as a model of human metabolic syndrome.
Yeah. Yeah.
So then you went and treated, you touched on this a little bit, but you treated the animal with the GLP-1 receptor agonist. And so you did tell us a little bit about why these drugs, but what were the gaps in the literature that kind of drove you to do this study?
Well, the gaps in the literature were, and I apologize. You asked me, Darlene, what you treat these mice with or humans. The first now approved drug is resmetirom, which is a TR beta, not alpha, but beta agonist. And this actually promotes both fat burning in the liver as well as attenuates the fibrosis, probably through, if you decrease the fat deposition and you burn more fat, then maybe that attenuates the inflammation and you get resolution. And that's, of course, was been shown in a clinical trial. So that's the first agent that's been used in humans for MASH. Now, it was interesting to us that our paper came out this month showing that GLP-1 receptor agonists, and we use dulaglutide or in this study, but the semaglutide has now been approved 2 weeks ago for liver disease and a MASH. The question is, how does it work? Because if you talk to experts in the field of GLP-1, they'll tell you that, well, there's some macrophages in liver that may have some receptors, but GLP-1 receptors are not in the liver. And then they also tell us, well, GLP-1 receptors are not in adipose tissue, but maybe they're on these few macrophages. So really, the big gap in the literature is how are these working to attenuate MASH? And so, as you probably know, because I think there's a lot of investigators out there listening to this who do liver studies. So how do you get steatosis? Well, 10% comes from what you eat. Well, in this study, we controlled what the mice eat. So they had the same amount of fat in their diet and they ate the same amount of food. About 25% comes from de novo lipogenesis. So the liver is getting fat and is saying, well, I'll make more fat and let it hang out in the liver. And then probably the rest, 65% or so, comes from lipolysis of adipose tissue. So here, well, how does the GLP-1 receptor work if it's not in liver or adipose tissue? And I think our contribution was, I think it's known that GLP-1 can attenuate de novo lipogenesis, but GLP-1 receptor agonists also, we found, have a major impact of decreasing adipose tissue lipolysis. Now, with both those important mechanisms being affected by GLP-1, I think this is a major ways that GLP-1 contributes to attenuating MASH. And as you know, many drugs are approved and we don't know how they work, but they help the disease. So I think what needs to be done as we go on from here is to understand if it's not working through GLP-1 receptors, are there brain effects? I mean, GLP-1 receptors are everywhere in the brain, as you know, more than just, I think, satiety centers. And they're being expanded to use for addiction, like smoking or drugs, cravings. The patients that I put on these drugs say, wow, I don't crave my sweets every night, or I don't need to go out and get a chocolate bar. So it really changes the patient's relationship to food. And I think it's through these brain mechanisms that are controlling not only behavior, but controlling metabolism in a big way. And so one of the things we hope to do is map the brain circuits into adipose tissue and find out which circuits are being affected by GLP-1.
That would be really cool.
Yeah. But I think that's where the mechanisms of how these things work is through the brain. And I think the brain really controls more of our body functions than we think, besides just our thinking and our activities.
Yeah. So, Will, I guess my next question is, you've touched nicely on the liver lipids, but you also had an atherosclerosis endpoint built into this study, right?
Yes.
Do you want to maybe touch on your findings there and what it tells us about GLP-1 receptor agonism in that aspect?
Oh yes, Kirk. Because we were, we do both liver and athero studies, and we've been doing the athero studies for many years. I've been doing them since 2000 was our first athero paper. So I fully expected the athero to be affected independent of weight. And we do on phos analysis where you take the whole aorta out and then you stain for like oil red O or a color, you stain for fat. And we absolutely, and these mice, as I said, get advanced plaques. We saw no difference in the pair-fed control mouse compared with the treated mouse. Now, I think we also reported in the paper, if you take the mouse and just give it the GLP-1 and don't weight control it, you do see improvements in athero. But now when you weight control it, to my surprise, we didn't see any difference. And of course, because we had a negative study the first time we did it a couple more times, and it was the same thing. And what we do in our lab is we have one person will be responsible for housing and feeding the mice. Then the fellows will be responsible for taking out the aorta, and then another blind person will do the aortic analysis, so they don't know who got what. So I have to believe that the athero was really highly dependent on weight, whereas the liver has GLP-1 receptor agonists have some specific effects to attenuate the liver disease.
Very cool.
Yeah, that I just want to put an emphasis on that. That pair-fed control group was really pretty important, right? I think it's
Yes.
something you can't really do clinically, and unfortunately, a lot of MAFLD papers don't actually use that. I mean, it's experimentally a little bit more complicated to use, but it is really important.
Right. Because someone has to come in at the end of every day and weigh the food to figure out what the GLP-1 mice ate. And then the
Right.
next day, that amount of food only is given to the pair-fed control mouse.
Yeah.
Yeah.
So there's more man work hours on that. Are
Right.
there any other key points or thoughts you'd like to leave us with as we close the interview, or maybe talk a little bit more about what you'd like to do next, moving this forward?
Well, we talked about doing more brain analysis to
Yeah.
figure out how the brain is controlling both metabolic changes. Lipolysis is a big thing in adipose tissue, how it's controlling lipolysis. And then is it also contributing to the metabolic changes like de novo lipogenesis and other things in the liver? So I think a big part of studying these agents is trying to figure out how the brain is working. And what I think also for the future, I looked up some future drugs that are coming by. So GLP-1 receptor agonists are like the base, the foundation. And
Yeah.
then, as you know, they're adding all kinds of other receptors. You know, we have the GIP, and that's a lot of interest in tirzepatide. Then they're going to add amylin to that. And then they're adding glucagon receptor agonist to that. So what I saw was that at least 5 different drugs are in phase 2 clinical trials, and at least 10 or more are in animal trials and
Yeah.
and going on to phase 1. So I think what we need, excuse me for saying this, is maybe artificial intelligence with good input to actually tell us, well, this patient has A, B, and C, and D, and this would be the best GLP-1 receptor agonist combination for patient A, B, C. So I'm hoping that we can individualize therapy as these different agents become available for human consumption.
Yeah, that's an amazing point.
Yeah, so I'm looking forward to the future of these agents and the future of studying adipose tissue biology.
Yeah.
Willa, we really want to thank you for joining us today and for sharing your expertise and discussing your recent work on weight loss independent effects of GLP-1 receptor agonisms on MAFL. I encourage the listeners to read the full article, which is the paper of the month and the cover. It's a beautiful cover in the September issue of Diabetes.
Thank you both. Bye now.
Welcome to another edition of Sweet Talk. I'm Kevin Williams, and I'm joined by our co-host today, Dr. Darleen Sandoval. Today, we're continuing our special series, which we're featuring recipients of the ADA's Pathway to Stop Diabetes Awards. These are early career investigators who are pushing the frontiers of diabetes research with creativity and bold new ideas. Our guest today is Dr. Alexander Nectow, a physician scientist at Columbia University. His lab studies how the brain controls feeding and metabolism, with a particular focus on a small but powerful structure in the brainstem called the Dorsal Raphe Nucleus, or the DRN. His pathway-supported research is reshaping our understanding of how this midbrain region regulates appetite, body weight, and ultimately risk for diabetes. Welcome, Dr. Nectow, and thank you for joining us today.
Thanks for having me. Really appreciate that introduction, Kevin.
Yeah, now we usually like to start off with a little bit of an icebreaker question. And so my question for you today is, outside of the lab, do you have any hobbies or interests that you enjoy that might surprise some people?
Lots of funny little hobbies. My favorite though is, I think my wife and I are huge fans of the New York Times crossword puzzle. So I think we're on our 168th, I think, today or something in a row. So it's been a bit of a hot streak.
Nice.
Nice.
Do you fill out the actual newspaper, or is this I know they have an app now with the New York Times mini crosswords and things like that. Do you do those, or?
Oh, we do the full crossword, but it's on
The
the app.
full
It's
crossword.
amazing. Yeah. Oh, it's awesome. Yeah. So we can keep track of it. We get those gold stars. You know, nothing like a good box checked off saying you've gotten something accomplished for the day.
Now, do you do them together, or is it a competition?
Oh no, we do them together. I think it
Do
would
them
be
together. Okay.
brutal
Yeah.
if we did competition. That
Yeah.
wouldn't end too well.
All right. Well, I get to ask you the first question, and let's kind of talk about how you got started. How did you first become interested in studying the brain's control of metabolism, and how did that lead you to the DRN? And then I would love to hear, if it's applicable, how you kind of weave your engineering background into your approach to neuroscience.
Sure. So yeah, a lot of great questions to unpack there. So in terms of I'll start with just how I got into metabolism and feeding. So in my PhD, I originally was interested in studying neurologic or neural control of behavior. And I'd end up joining Jeff Friedman's lab, who of course cloned leptin, and I'd spent many years thinking about these problems on many different levels. And while in the lab, I just got, I started thinking about a number of different ways to tackle neural circuits that regulated behavior. And in Jeff's lab, a natural connection was metabolism, in particular feeding behavior. So working with Jeff, we, try to sculpt out a number of different areas that would be interesting. And the Raphe that I'll talk about in a bit, I'm sure, became one of those areas that I'd found was really clearly playing some kind of important role in regulating appetite and metabolism more broadly, but something that a lot of people hadn't tapped into in the way we've been thinking about it. So that was kind of my entree into the field of neurometabolism. And then in terms of the engineering mindset, as you mentioned, I trained as an engineer. Even while I was an engineer, I was very interested in systems neuroscience. So a lot of our early work on applied mathematics really worked on trying to solve different ordinary differential equations that applied to modeling neural circuits using the Hodgkin-Huxley-like ordinary differential equations. And in terms of the engineering background, I really wanted to bring that to my PhD, and that's what got me into doing a lot of this tech development. So Jeff's lab had implemented a number of techniques using ribosomal tagging that was based off of the pioneering work of Paul Greengard and Nat Heinz, who were also at Rockefeller developing these ribosomal tag-based approaches at the time. So I got really into kind of doing more molecular engineering to develop approaches where we could tag the ribosome and pull it down in neurons that were projecting to a certain target region or expressed its own unique molecular milieu.
Nice.
One thing I did not mention in the intro is that you contributed a perspective article this month in Diabetes, and that article really is focusing on a lot of the work that was generated from the Pathways Award. And in that, you emphasize the DRN, doesn't just house hunger or satiation related neurons, which I think we're going to get more in detail into those neurons in some follow-up questions, but it also sits at a crossroads of many inputs and outputs across the brain. And so I was curious, as a global perspective of the dorsal raphe, do you think it's best thought of as an integrator hub that brings together various metabolic and behavioral signals, or is it more specialized in driving specific aspects of appetite?
Yeah, it's a really great question. So I think our work has specifically highlighted the appetite regulating features of the raphe, but I think even work from my own group, as well as those of many others in the field, it's become clear that the raphe is very much an integrator. This is something that had been thought of for quite a while, given the role, given it housing the single largest population of serotonin neurons in the brain. And given, as you mentioned, its input-output connectivity. I mean, we know that this region is receiving inputs brain-wide and then sends reciprocal projections back to many of those structures. So I think the raphe is playing more of an integrative role across many different behaviors and physiologic processes, not simply food intake or energy expenditure for that matter even.
So your lab has identified specific subpopulations of DRN neurons, and some of those actually drive hunger, like GABAergic neurons, for example. So can you tell us a little bit about those studies and what you learned from them?
Sure. So about 15 years ago or so now, when I was in Jeff Friedman's group, we had been performing some basic just FOS studies, so labeling of the raphe in different metabolic challenges, one of which being hunger, for example. So we'd starve an animal for overnight and look to see which regions the brain lit up and where in those regions we saw activation of neurons. FOS, of course, being a surrogate marker for neural activity, so that could help us pinpoint exactly where cells were being activated. So one of the regions we found was the dorsal raphe nucleus, which had validated a lot of our suspicions at the time and also some other findings from the literature. And just based on the colocalization, or the localization rather, of these FOS cells, we hypothesized that these were GABAergic neurons because they clustered medially but avoided the midline, and then they kind of branched out into the more lateral aspects of the raphe, or what were classically called the wings. So performing some different functional manipulations using optogenetics and chemogenetics, we were able to establish that these GABAergic neurons were promoting food intake. And more recently, we've found that these neurons probabilistically drive food intake. And we only were able to find this out when we actually started physiologically stimulating these neurons. So more recently, we've been recording from them and finding that they're actually active for very short windows of time, which was quite surprising to us. And by mimicking that stimulation, we found that we can induce these straight transitions quite rapidly. So we're very excited about understanding how these GABAergic neurons really regulate food intake. And it seems to really promote the beginning of a meal rather than the middle or the end.
Interesting. And do these neurons regulate other aspects of energy homeostasis, or is it really more centralized to feeding control?
So that's a really great question. Right now, we don't have the degree of molecular control or genetic access into the neurons that I'd like to have. So we've been studying these GABAergic neurons and assuming that they're this broad homogeneous population, when in reality, they're comprised of at least 3 different subtypes that we published on earlier this year. So we're just now diving into those subpopulations to establish which of them might regulate different aspects of behavior and physiology. We know for certain that, again, a subset of these neurons definitely controls appetite. We think that there's a non-overlapping subset that then regulates energy expenditure by suppressing thermogenesis. So while these neurons in aggregate are definitely controlling various aspects of energy homeostasis, it seems like there is some kind of cell type specific mechanism whereby this region could independently regulate food intake and energy expenditure.
Interesting.
And in that previous question, you were highlighting some stimulation of feeding subpopulations of neurons. But on the flip side of this, you've also identified or suggested that there are some neurons within the DRN that are suppressing feeding, with the particular maybe emphasis on the CCK expressing cell types. And in that, it's highlighting those neurons seem to be acting more as detecting a running tally of number of bites that the animals are eating, and they're active during eating, but it's only kicking in later to help decide when to stop a meal. And I was curious, why do you think on a global scale the brain kind of evolved this delayed stop signal instead of shutting down more rapidly feeding behavior?
Yes, that's a great question. A few different things to unpack there, but I'll just go to the final point there, which is really why did the brain evolve these different mechanisms? And I think if you look at the raphe in particular, it's kind of a simulacrum of a hypothalamus, or a hypothalamus in miniature that sits in a much more evolutionarily ancient structure. I mean, these are brainstem neurons that are conserved, from, all the way down to zebrafish and probably lower organisms, and all the way up to primates and like humans, for example. The raphe has to triage a lot of information, right? I mean, as we were discussing before, and you mentioned, it's really an integrator. So it has to take a lot of similar classes of information across many different molecularly differentiated cell types and leverage that information differently in space and in time to control different aspects of behavior and physiology. So in the case of our recordings of these neurons, what we've been finding over the past few years in both published and unpublished work is that they seem to react in terms of their sensory properties, different stimuli classes, rather similarly. So for example, these GABAergic neurons that we study that promote the onset of feeding respond almost exactly as the CCK neurons do, at least on short time scales. And again, they have to leverage that information at different points in time to make specific decisions about feeding. So these GABAergic neurons need to act rapidly to drive the onset of feeding, whereas these CCK neurons need to use similar classes of information to act at a temporally distant point in time. What we're now learning by studying, different subpopulations in the raphe is that, again, they're just leveraging the same types of information to make relevant decisions in time about when to start and stop eating. And it's likely that this extends to many other behaviors and physiologic processes. We just haven't really, dived too deep into that just yet, but we're certainly interested in it.
So to follow up on this idea of the DRN being an integrator, what do you know about how it communicates or works with the hypothalamus, the NTS, the PBN to regulate overall energy homeostasis?
That's a really great question. So, I mean, from a lot of our studies that we've done, a lot of the loss of function phenotypes we're seeing are fairly decent, but they're not like that of disrupting a sensory locus, like the NTS, for example. So we're not completely shutting down communication to the forebrain when we manipulate the raphe. So it's very much a modulatory center, very much, which is in line with classic thinking about the raphe and serotonin's role in particular in modulating different forebrain structures and forebrain dependent functions for that matter. But I think that the Raphe clearly is important in triaging these different classes of information from the hindbrain, particularly the pons, the medulla, and either shunting that information or relaying it to the forebrain, or just turning around and actually leveraging it to make shorter-term decisions about feeding, whether that's on a bite-to-bite basis or meal-to-meal basis. I'm not sure yet. That's something that we're really excited about looking into. But it's definitely one of these more modulatory centers than I'd say than like a primary locus as the seed of motivation for appetite. I mean, we know a lot about hypothalamic structures and their role in initiating these different behaviors and motivating them over longer time scales. I don't think the Raphe does that. I think it's doing something much more nuanced that links forebrain and hindbrain signaling.
Cool.
And as we had mentioned, you are a recipient of the ADA Pathway to Stop Diabetes Award, and you've written this perspective highlighting the work involved. I was curious if you could give any insights to how this support from the ADA might have shaped the trajectory of your lab and allowed you to dig into these questions about the dorsal raphe.
The ADA has been, I give them a ton of credit because they were honestly the first major supporter of my work when I started my laboratory as a fellow when I was in Princeton. So at the time, I was actually simultaneously running my lab and going through clinical training here now at Columbia, where my lab is currently. So through that time, the ADA actually supported me through two different grants, through the Pathway mechanism as well as the Innovative Basic Sciences program, and really allowed my lab to grow in size and in breadth in terms of what we were studying. The project that was funded through the Pathway program funding mechanism focused on these GABA neurons, but because of that, because we were funded through that, we were able to really branch out and understand not just these GABAergic cell types and the regulation of appetite, but a number of other different populations that we've recently published on and that we're really excited about. So I think that the ADA was absolutely, and the Pathway funding mechanism was instrumental in launching my career and allowing me to continue working as a clinician scientist while driving this work forward.
Is there any advice that you would give to some early career scientist that might be thinking about applying for this type of award or a similar award, and how to approach the funding mechanism and going through the process?
As a clinician scientist now, I think I have a bit of a different perspective on it than I did maybe over a decade ago when I hadn't yet kind of embarked on this journey, I guess, this clinical training journey. But for me, the big thing is that our job as scientists is to drill down the basic science and the fundamentals, whether in terms of investigating physiologic or pathophysiologic mechanisms. And the ADA, like many other foundations, is very targeted and really wants to understand mechanisms governing diabetes and identifying cures for that in the future, or at least effective treatments. So I think, having a really targeted proposal in terms of explaining how the biology could link to the pathophysiology is really important. And that was something that we always kept in front of mind when we were writing these grants. We were studying a novel population of cells that we knew played an important role in regulating appetite and potentially body weight. And we wanted to be able to link that to glucose homeostasis or dysregulation of those processes through these neurons. So that was something that I would really recommend if people are applying for these types of awards, is to think about the pathophysiology.
So you mentioned one thing you were excited about in my previous question, but what are some other big unanswered questions, either about the DRN or appetite regulation as a whole, that you are most excited to tackle? And are there any new technologies that you're really excited about to try to address that?
One of the things that we're really trying to pin down now is figuring out what this region is actually integrating. We keep talking about integrators, and that's a nice buzzword, but really, what does that mean for the region, and what does that mean for behavior and physiology? So one thing we're really interested in is understanding state transitions, which again, I've kind of hinted at this throughout our chat, which is that the cell types that we've identified really seem to play an important role at transitions, whether the onset of a behavior or physiologic process or the offset of a behavior like feeding or a related physiologic process. And why that really excites me is that this is something that's highly conserved, again, down to the level of zebrafish. So there are a number of groups that have done whole brain recordings looking at state transitions and really exploration exploitation paradigms in the context of feeding. And they've found that the raphe is always really the most active or highly correlated structure in terms of these functions. So we'd like to understand why that is. I mean, everything we find really does echo these studies from, organisms from zebrafish on up. So we're really excited to parse that out. I think the technologies that we're really interested in using these days are, more high throughput, using molecular profiling in situ, so we can look at the cells that we're studying, whether on their own or after doing imaging studies, and then doing a lot more in-depth imaging of these different circuits at scale, so we can understand really what these cell types are doing and who they're interacting with while they're, active.
That's really interesting.
Thank you, Dr. Nectow, for joining us today and sharing your ADA pathway-supported research and insights. And thank you to our listeners for tuning in to this episode of Sweet Talk. Before we go, Dr. Nectow, could you let our listeners know what you have coming up or where they can learn more about your ongoing work? Do you have a website? Are you on the socials?
So I like to think that I've made my career by staying off of social media for the most part. But yeah, any time you'd like, you can reach me through my lab website, nectowlab.org. I am on Twitter. I guess it's now X. I'm at CPNoct, NOCT. So yeah, you can find me in either of those places.
Perfect. Be sure to check out Dr. Nectow. We also want to remind our listeners to check out the Perspective article on Dr. Nectow's pathway-supported research titled The Dorsal Raphe Nucleus and the Integrative Control of Feeding, which is featured in this month's issue of Diabetes. As always, don't forget to like, follow, and subscribe for more diabetes bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.